Hello and welcome back. Welcome to today's lesson. We're going to look at asexual reproduction in plants. Last week, we began to look at sexual reproduction and we compare asexual reproduction to sexual reproduction. So, in this lesson, we will focus mostly or entirely on asexual reproduction. So, to begin, what is asexual reproduction? In asexual reproduction, it is a process in which new organisms are produced from a single parent without the involvement of gametes or sex cells. When we looked at fertilization in the previous lesson, we looked at sexual reproduction. And in sexual reproduction, you have two parents. The male will produce the male gametes and the female will produce the female gametes, and these gametes will fuse together. However, in asexual reproduction, there is no need for these gametes. So there are two types of asexual reproduction in plants. We have natural vegetative propagation and we have artificial vegetative propagation. And as these terms suggest, the natural vegetative propagation that occurs naturally without the aid or without the help of human beings. Whereas artificial vegetative propagation, these are done through the intervention or through the assistance of human beings. So on this table, we have some examples of vegetative or natural vegetative reproduction. So we'll go through the vegetative organs. We'll quickly go through this table. We have the sucker, and the sucker is an underground shoot growing from the basal part of the parent plant. Example of sucker, a good example of it would be banana. Another example could be a plantain, and pineapple is also considered as a sucker. Then we have tubers. We have two types of tuber. We have a stem tuber and we have the root tuber. Example of stem tuber will be yam and Irish potato. An example of root tuber will be the sweet potato and cassava. Next we have rhizome. The rhizomes, these are thick underground stems. An example, very good example of rhizome would be the ginger. Some other examples are listed here. We have the corm. What is the corm? It is a short, swollen underground stem. A typical example would be arrow. Next, we have bulb. So the vegetative organ is called a bulb. It is the leaf. It is mainly the leaf of the plant that is referred to as a bulb here. An example would be onion. Next, we have stolon. These are slender, creeping stems. The stem will be growing on the surface of the earth. Example, we have the Bahama grass and some other grasses that we may have that are called stolon. And then we have runners. These are also creeping stem and they're usually on the soil surface. Again, the difference here, let's go back a little bit. The stolon, the difference between the stolon and the runner. The stolon, as you can see in the description, is an underground stem that is creeping underground. Whereas the runner, that is found on the surface of the soil. An example would be strawberry and pumpkin. All right, so that's just a quick look at vegetative propagation, natural vegetative propagation. Now we'll go into artificial vegetative propagation. There are different artificial vegetative propagation, and we'll look at a few of them. First one that we'll look at here will be cuttings. So for cuttings, pieces of stem, it could be pieces of root as well, or leaves, are taken from certain plants and then they're given the correct conditions. They're given certain conditions and they're allowed to grow. Example of plant parts that you can take for cuttings would be guava, cassava. Now we just looked at cassava as an example of a root, a tuber. However, part of the stem can be taken and that can be used for cuttings. And we have sugarcane and pomegranate. The part that is used, that is removed, it's provided with suitable humidity, suitable environmental conditions, for example, light and suitable temperature, and these conditions will allow proper growth of the cuttings. They are provided, there are two types, there are two types of cuttings, and we have the stem cuttings and we have root cuttings. So we'll go through the stem cutting in a little bit more details now. So the cuttings are taken early in the morning when the cells are turgid. We looked at osmosis, and from that you would have been introduced to the term turgid. So turgid simply means that the plant has taken up a lot of water, and the cells have a lot of moisture or a lot of water in it, and they're turgid. Example of plants that we use for cuttings, for stem cuttings, would be citrus and sweet potato. After removing the cutting, it is normally wrapped in a moist tissue because you don't want the cutting to lose moisture. This may not be necessary if you're going to use the cutting immediately, but usually it is not being used immediately after it is removed from the parent plant. The base of each cutting is trimmed. You would use a sharp instrument and trim the base of the cutting, and the leaves are usually removed or partially trimmed. They're usually completely removed. The base can be dipped into rooting hormone. Now, the rooting hormone cannot be used on any and every plant. They're not very safe on all plants. So you place the base of the cutting into a rooting hormone. Some plants, however, do not require rooting hormone for them to actually grow new roots. A propagator is prepared, and the propagator will have suitable garden soil, suitable potting soil, and this will allow the growth of the plant to occur very easily. The propagator soil should be watered and should be placed in an area where there is suitable amount of light and suitable temperature for it to grow properly. So this video is demonstrating how cuttings are being done. So here we have the cutting already being extracted from the parent plant, and the base is being trimmed with a sharp instrument as mentioned in the previous slide. So a sharp cut is being made on that cutting. So now we have the stems and they're ready for the cuttings. And next we prepare the soil. The soil should be kind of loose, so it should not retain a lot of moisture. And then you place the cuttings into the soil. Now, if you have a rooting hormone, you can place it into the rooting hormone, the stem cutting, you can place it in the rooting hormone and then place it in the propagator. But in this instance, it all depends on the type of plant that you're using. In this instance, it was placed directly into the soil, into the propagator. Propagator is really the area where the plant will grow. And then you add water to it and you expose it to suitable growing conditions. And then you will eventually see the growth of new branches, and you will see new leaves developing at the side. So after 38 days, this is how it may look like. Different plants will have different growth patterns. Next, we have root cuttings. So here, pieces of root are taken from the parent plant. A sloping cut is made at the base, and the top of the root cutting remains at the soil level. And an example is shown on the diagram. The entire root structure is placed below the surface of the earth, and the top of the cutting is at the soil level. It doesn't go above the soil level. The cutting is placed into rooting medium, just like how we place the stem into rooting medium. And the root is taken from the parent plant usually before the rainy season begins. Next, we have budding. Budding is a form of grafting. Again, we're looking at artificial vegetative propagation. We just looked at cuttings. There are two types or two ways in which cuttings are done, or two parts of the plant that are taken for cuttings. We have the stem cutting and we have the root cutting. Another method is budding. So it's a form of grafting. Some persons may use the word grafting interchangeably. So a single bud, and you refer to that as a styon, is removed from one plant and it is inserted into the stem of another plant, which is called the stalk or the root stock. It is commonly used in citrus plants, especially in Guyana. Example, lemon, lime, and orange or tangerine as well. Two parts are mainly needed for this procedure. You need the budwood, and the budwood is a stem that is removed from a plant that has the desirable characteristics. So for example, if you have an orange tree and that orange tree is bearing fruits that are very sweet in taste and very good large size of orange, very good produce you're getting from that orange tree, well then that could be the source or the parent plant where we collect the budwood. The budwood should not be from a stem that is too old, nor should it be from a stem that is very young. And next, you need the rootstock. And the rootstock is the plant where you will add the bud to. So the procedure, let me go through the procedure quickly. What is needed is you need a budding knife, or any other instrument can be used, but it's preferable that you use a budding knife because the budding knife have features that would help you to carry out this procedure more easily. So the budding knife need to be sterilized. Locally, what you can do is to add bleach into the water and place the budding knife into it to sterilize it. Sterilizing the knife is actually to remove disease-causing organisms, to remove fungus, to remove bacteria, and so forth that may affect the plant that you're trying to bud. Then you remove the side branches from the rootstock. So the rootstock is the root. So you remove the side branches from the rootstock and you cut at the top of the rootstock. So you leave about 30 cm of the rootstock, 30 cm from the soil, and you remove the top layer of the rootstock. A T is then made, or we may have an inverted T as well. This is made on the rootstock using a budding knife, as was shown earlier. The scion is then cut from the budwood, and this is the bud that you're removing from the budwood, and then it is inserted. So the scion or the bud is inserted into the rootstock to the area where you made that T. Then a tape is used, usually a polythene tape, and it is wrapped around the bud and is covering the bud that you just inserted into the T that was made. After taping the location just above the rootstock, you would cut off that section. And then after about 7 to 10 days, the wrapping can be removed to allow the bud to develop into a new stem. So the following here is a demonstration of how budding can take place. So we have here the rootstock. And as you can see, an incision is made, a T is being made. A bud knife should be used, a budding knife should be used for this procedure, but in this instance, the individual was using a razor blade. Next, the bud is being removed from the budwood. So we have a nice piece of bud there, or scion. And then the bud is inserted into the rootstock. So it fits nicely there. And then the two parts will eventually go together and a new stem will emerge. So that's how it would look after it is taped. Usually the area where the bud is exposed is not taped and the surrounding area is taped using the polythene tape as I mentioned before. So that brings us to the end of this short lesson. If you have any questions regarding the procedures that were explained in this lesson, do not hesitate to ask your questions. Thank you for listening once again. Until the next lesson.